So when the pandemic first began, we had to kind of repurpose our lab to stay involved. And what we were able to do is look at the kind of work we were already doing with these oncolytic viruses, which are already given to patients who are immunosuppressed and is used to treat cancer. And we thought that they were the perfect platform to help develop a vaccine with. So we quickly decided to try and make a COVID-19 vaccine using the same strategy that we were using already for targeting cancer.
And so you're using a virus-based vaccine. What's the advantage of a virus-based vaccine?
Yeah, so most vaccines have two parts. They have the part that's recognized to try and train your immune system against the disease in question, and then you have another part called the adjuvant, which is help to create an overall immune response. And this is important for just getting good overall immunity against whatever target you want. So viruses are really good at doing that because it mimics most of the things that we're doing in terms of virus infections like, say, SARS-CoV-2. So by doing this, we get really good antibody responses, T cell responses, and this boosts overall immunity.
And so you tested how did you test this?
We used a few different methods. We tried to go with some of the standards that were being used in the field. The big one is looking at the antibody responses. So we try to quantify how much antibodies our vaccine is producing and also confirm that these antibodies can neutralize the virus itself. So we created these fake viruses we call pseudovirus, and we show that the antibodies that are created from our vaccination is able to prevent the virus from infecting new cells. And then we also try to look at T cell responses, which are another part of the immune system that's really important for giving long-term protective immunity. And we use some other methods again to make sure that the T cells are becoming activated with our vaccine.
And so you tested them both in mice and in monkeys?
Yeah, that's correct.
Why was the replicating virus better than one that was non-replicating?
So one of the issues with non-replicating viruses, the advantage is that they're very safe, but the problem is that they typically only get one round of infection, and this can, when you're trying to express the foreign antigen to create immunity against, you only get so much expression of that protein in that case. Whereas if you use a replicating virus, it can continue to create that antigen over and over again, so you get more of it. It allows you to create a better antibody and T cell responses because you're getting longer, more prolonged expression of that antigen.
The study notes that this would be potentially really important for global vaccination. Why is that?
So one of the things is that it's still tricky to give vaccines all around the world because some of the RNA vaccines are not the most stable. They require special refrigeration. The thing about these pox-based vaccines is that they're very stable. We did a few tests within the study where we show that we can keep the virus at room temperature for over a week and it's still fully functional. And I think this will help with distribution and also production. It's very easy to manufacture on a large scale for this virus.